Hi, thank you for your interest in MiDOG services. After we process and analyze the samples we received, our vets receive a comprehensive and detailed report outlining the results of the sample via email. When you click on the link that we sent you, you should open a report that looks something like this. This video is going to help you navigate through the report while explaining the terminology and providing interesting insights so that you can make the best decision for your patients. The first section of this report is the metadata that we received from our vets. The first column here contains the basic information about the pet. Second column here has information about the vet, and the third column has information about the sample type and the health status. The MiDOG report has three important parts. The first part is the clinically relevant pathogens that we detect. We detect both bacterial and fungal pathogens in the sample given to us. Here we have results for our pathogens detected in this sample. The first column here lists all the microorganisms that we detected. All of these organisms are active links, so when you click on these links, it should take you to a Google search for these microorganisms. The second column is the absolute abundance of the species detected. Absolute abundance is the cell count for the species in the sample. So for example here, alkali genes faecalis, we see 72,000 cells of the species in the sample. The relative abundance is the percentage of the specific species in the bacterial community of the sample. So here we see 26.4% of alkali genes faecalis, which means that 26.4% of the bacterial load in the sample contains alkali genes. The next section here is the analysis of total bacterial and fungal abundance in the profile. This is the ratio of total bacteria versus fungi in the sample that we processed. This section is very useful to help us identify if the sample has a potential bacterial infection or fungal infection. The next section is the antibiotic resistance genes detected for the relevant pathogens that we identified earlier. Here we list the antibiotic resistance profile for all of these pathogens, and as we can see that most of these pathogens are susceptible to the antibiotics listed here, except for the first one, which is alkali genes, which is resistant to sulfonamide. We also provide the suggested dose and the drug delivery method for these antibiotics. The next section is provides additional information about the sample, which could be clinically relevant findings. In this, we talk about the microbial diversity score of the sample that we received. The microbial diversity score basically summarizes how many different bacteria is present in the sample. So if the diversity score of a sample is below our clinically healthy range, that indicates an unbalanced microbiome. Our clinically healthy range has been developed over the years from all the healthy samples that we've received. The next section here are the interesting bacterial and fungal microbes that we detected. These microbes are interesting because the relative abundance detected for these microbes are usually seen outside the clinically healthy reference range, which is why it's listed as an interesting set. The next section is the complete bacterial profile of the sample in comparison to the clinically healthy range. We also list the different bacteria that were detected in the sample along with the absolute abundance, its relative abundance, and the relative abundance seen in our healthy range. We also have a similar fungal analysis, which lists all the fungal species detected in this sample along with the clinically healthy reference range. We also provide references to the pathogens that we have listed before, and along with the method section of our method for our technique. If you have any further questions, please contact us at info@midogtest.com or call us at 833-456-4364. Thank you.